Our next lab protocol is called ELISA, which stands for Enzyme-linked Immunosorbent Assay. As you might guess from its name, ELISA is an immunologic technique, which uses specific antibodies or antigens to detect the presence of a target that you're interested in. There are two types of ELISA, direct and indirect, and both are typically done on a microtitered plate like this one, and you can test a different sample in each well of the plate. For a direct ELISA, we want to see if an antigen is present in a sample, for example in our blood or serum. So let's say we want to know if and how much of an antigen represented by this orange star is present in our serum. First, we put the whole sample into a well in our plate, and then fix the antigens to the bottom of the well. Then we add some antibodies that are specific to our antigen of interest, which again is the orange star. We want enough antibodies to make sure to hit all of our target antigen that's present if we want this assay to be quantitative. Then we wash the plate, and all of the antibody that isn't bound to an orange star is washed away. Since this antibody is linked to an enzyme that can generate a color, we simply add the substrate for that enzyme and it will generate a colored product. So if our antigen wasn't present in the original sample, we wouldn't see any blue color here. And if there's a little bit, we'd see a little bit of color, and if there's a lot, we'll see a lot of color. We can then quantify how much a color there is to figure out how much our original antigen was present in the sample. For an indirect ELISA, we can either look for an antigen or an antibody. Let's start by looking for the same orange star as before. The first few steps are the same, but there's just one more step here. In this case, our primary antibody doesn't have an enzyme attached to it. Instead, we can now add a secondary antibody, which is specific for the Fc part of our primary antibody, and then wash away the excess. The secondary antibody has the color-generating enzyme, and we can use it to generate a color and quantify the original amount of antigen just like before. So why would we do indirect when we could save ourselves a step by doing a direct ELISA? It's mostly a matter of convenience because it's easier to have a stock of labeled antibodies that target the Fc portion of a wide variety of other antibodies, like these secondary ones, rather than have to put a label on each primary antibody you'll ever use. Okay, one last thing. So how do we determine how much antibody is present in a sample? Let's say we want to know if this orange antibody is in a sample. We have to already know what its target is, which in this case will be the black star, and we have to start by having a well with that target antigen fixed to it. Then, kind of like before, we add our sample, which of course has a variety of antibodies, but when we wash it off the plate, only our antibody of interest will be left behind. Now, just like before, we can add a secondary antibody which targets the Fc portion of our antibody of interest, which is linked to a color-generating enzyme, and by measuring the color, we can infer if and how much of our orange antibody was in the original sample. So when you're done, you might end up with a plate that looks like this, if the sample is towards the bottom of a higher concentration of the antigen or antibody you're testing for. And the plate can be read by a plate reader to tell you exactly how much signal you're seeing. ELISA tests are extremely sensitive, so they're often used to screen for a disease such as HIV. Since false positives are somewhat common, these tests can be confirmed by a more specific test, such as a Western blot. So Western blots and ELISA both use antibodies to detect a target antigen. But can you think of why a Western blot might have better specificity? One reason is that the Western blot allows you to observe the size of what the antibodies are sticking to. So if they stick to something that's the wrong size, you'll realize the test is a false positive, whereas with ELISA, all you know is that it's stuck to something, but you have no other information about what you're detecting.